Hello everyone, welcome to today's session. Today we are going to see Class 12 Biology, Chapter 8 Microbes in Human Welfare. Have you ever wondered how microbes can help us make products on a huge industrial scale? We usually think of microbes as disease causing organisms, but actually, microbes are extremely useful to humans. So, they are used to produce beverages, antibiotics, organic acid, enzymes, and important medicines. But we cannot grow microbes in a small laboratory flask, but we need a production on a huge scale. So, let's see the next one. So what do industries use then? They use the fermenters. Yes. So the fermenters, what are fermenters then? Fermenters are very large vessels in which microbes are grown on an industrial scale. So here you can able to see the fermenters. Yes. So I'll ask you one question. See, if a factory wants to produce a microbial product in a very large quantity, will they use a test tube or a fermenter? What's your answer? Yes, it's fermenter. Very good. So next we are going to see the fermented beverages. See now, this is the oldest use of microbes, children. So it was producing the beverage. So this is the our star player who is the Saccharomyces cerevisiae. So you may already know this yeast from bread making. So we have studied this in our previous chapter, right? So the same yeast is also called as the brewer's yeast. So it is used to ferment the malted cereals. It is used to ferment the malted cereals and the fruit juices to produce ethanol, right? Okay. Just to tell me, the same yeast is used for bread and beverages also? Yes. See, during fermentation, yeast converts the sugar into ethanol and carbon dioxide under suitable conditions. Okay, then what about the distillation makes the difference? Yeah. See, why are wine and beer different from whiskey, brandy, and rum? Can you tell me? See, wine and beer, they are produced without distillation process, but whiskey, brandy, and rum, they are produced after the distillation process. So here is one exam tip for you. The same is Saccharomyces cerevisiae. Yeah, both races bread dough as well as the ferments beverages. Yes, it's a very important thing for your CBSE examination. Next, we are going to see the antibiotics. So this is the one of the most important discoveries which is involving microorganisms. Yes, that is antibiotics. See, what is an antibiotic? An antibiotic is a chemical substance is produced by some microbes that kills or retards the growth of other disease causing microbes. So it kills or retards the other disease causing microbes. Do antibiotics kill viruses? Can you tell me? No. See, antibiotics are used against bacterial infections only. It cannot kill viruses. So next we are going to see the Fleming's chance discovery. Okay, now I'll tell you one fascinating story. Yeah, Alexander Fleming, he was working with the Staphylococci bacteria. So here you can able to see, yes, he was working with the Staphylococci bacteria. So one day he noticed something unusual. A mold had grown on an unwashed culture plate, okay? And around the mold, the staphylococci were not growing. You can able to see here, no growth was here. So Fleming, he realized that the mold was producing a chemical that stopped the bacterial growth. And he named the substance as the penicillin. He named the substance as the penicillin. After the mold, Penicillium notatum, okay? So look carefully here. So here is this is the place where the bacteria is not growing, right? Okay, and this clear area is called the zone of inhibition. But after the Fleming's discovery, see, it was not the end of the story, children. Then what happened? Ernest Chain and Howard Florey, yeah, they later established the full potential of this penicillin as an effective antibiotic, and it was extensively used to treat the wounded soldiers during the World War II. And also, because of this discovery, Fleming, Chain, and Florey, they shared the Nobel Prize in the year 1945. Yes, and also here's one example for you. Antibiotics have helped control plague, whooping cough, diphtheria, and leprosy. Okay, let me ask you some question. You try to answer. So who discovered penicillin? What's your answer? Alexander Fleming. Who established its full potential? Ernest Chain and Howard Florey. And when they got the Nobel Prize? In which year? 1945. So next we are going to see the chemicals and enzymes. See, microbes are not only useful for beverages and antibiotics, they are also tiny biochemical factories. Yeah, they are also tiny biochemical factories. See, they can produce the organic acids, they can produce alcohols, enzymes and other useful molecules. So first one we are going to see the organic acids from microbes. When okay, let's see this one. So Aspergillus niger, it gives citric acid. Acetobacter aceti, it gives the acetic acid. And Clostridium butylicum, it gives the butyric acid, and lactobacillus gives the lactic acid. So just I'm going to give you the product. Now you tell me the microbe. Yes, if I tell you citric acid, what you'll say is Aspergillus niger. If I say acetic acid, Acetobacter aceti. Lactic acid, lactobacillus. Butyric acid is Clostridium butylicum. Yes, you should be thorough with all these things, children. Yes. And the next we are going to see the microbial enzymes. See, microbes also produce commercially useful enzymes. For example, let us talk about the lipases. See, lipases are used in detergents. Why they are using the lipases in detergents? Because they help to remove the oil stains from the clothes. Now just think about the bottled fruit juice. See, why does a bottled fruit juice often look clearer than juice prepared at home? You tell me. Your mom will prepare the juice at home, no. But the juices which we get from shops, it is much clearer than the juice prepared by your mummy, right? Why it is so? Because of the pectinases and proteases. See, these microbial enzymes, they help to clarify the fruit juices, right? So remember all these things. These are very important for your board examination. Next, we are going to see the bioactive molecules. See, now we have some very interesting microbial products. Yeah. So microbes can produce molecules that are useful in medicines. So these three names are especially important for your examination. One is the streptokinase, cyclosporin, and another one is the statins. Yeah. So let us start with the streptokinase. So it is produced by the bacterium Streptococcus. It is produced by the bacterium Streptococcus and modified by genetic engineering. And it is also used as a clot buster. It is used as a clot buster. So how we can say, just imagine blood vessel becoming blocked by a clot. So the streptokinase, it helps to remove the clot in appropriate clinical situations. Okay. And even it is associated with blood vessel clots in patients who have undergone the myocardial infarction. Okay. So next we are going to see the cyclosporin A. Yeah, it is produced by the fungus. It is produced by the fungus. What is a fungus? Trichoderma polysporum. And also it is used as an immunosuppressive agent in organ transplant patients. Okay. Let me ask you one question, children. Is cyclosporin A associated with increasing immune activity or suppressing immune activity? Suppressing immune activity is the answer. Next one, we are going to see the statins. So this is the last one. Okay, it is also produced by the yeast. And what is the yeast? Monascus purpureus. So they are used in blood cholesterol lowering agents. Okay, they are used as blood cholesterol lowering agents, and they work by competitively inhibiting the enzyme which are responsible for cholesterol synthesis. Okay, so we have come to the end. So I have given you, as usual, I have given you all the points, yes, and important topics, everything I have given you. Let me ask you question. You just try to answer these questions. So all possible one mark question. So industrial vessels used to grow microbes. What you'll say? Yes, fermenters. And yeast used in making bread and beverage fermentation is Saccharomyces cerevisiae. And the first antibiotic discovered is penicillin. And who discovered it? And Alexander Fleming. And penicillin came from which mold? Is Penicillium notatum. and citric acid, Aspergillus niger. Enzyme used for oily stains are lipase, and enzymes used to clarify bottled fruit juices, pectinases and proteases. Clot buster, streptokinase. Cholesterol lowering molecule is statins. Yes. So we have come to the conclusion part. Watch the full video. Yes, don't skip it. We'll see you in the next video. Thank you.